Let's look at the practical aspects of how viruses can be grown in the laboratory. This is often required for diagnostic purposes or research studies to advance knowledge on new viruses, and also the manufacture of vaccines. Unlike bacteria, viruses need host cells to grow. Some animal viruses, not all, can be grown in cell culture. These use continuous cell lines which are stable when grown in flask or tissue culture plates, so they can be maintained indefinitely. They are different to primary cell lines which are taken directly from humans or animals and tend to die after only a few generations. Continuous cell lines can be cultured in the lab for long periods of time if they are kept in a sterile environment. Most are derived from mammals such as humans and other animals and have been transformed in some way. Some are from malignant cancers or tumors. The diagrams show some cells in culture. The tissue culture plate on the left contains both normal and transformed cells. The transformed cells are red and the normal cells are blue. After culture, for some time, the transformed cells continue to grow and multiply, whereas the normal cells die. The propagation of large amounts of viruses, usually for making vaccines, is facilitated by inoculating living animals or eggs which have been fertilized so the embryos are present. This is the conventional methods for producing vaccines, such as the seasonal flu vaccine. That's why previously some adverse reactions occurred during vaccination in individuals who have allergies to egg proteins. Today, these allergens are removed to ensure that egg allergies are not an issue. In this half of the lecture, we'll shift from virus multiplication to virus identification, which is required for the diagnosis of viral infections. This is quite a list of techniques, and we'll go through each one of them in detail. These are tests that are used often in many virology diagnostic labs, and so I think it is essential to be familiar with them, as they'll help you interpret the results of these tests, as well as have a better understanding of why and when some of these tests are used. Since viruses are so small, electron microscopy is used, traditionally for imaging and identification of viruses. It is not possible to visualize them using compound light microscopes, as the magnification is insufficient. There are two types of electron microscopy: transmission electron microscopy and scanning electron microscopy. Transmission EM has higher resolution and magnification, and most of the images of viruses are captured with transmission electron microscopy, whereas scanning EM is used more for imaging host cells that have been infected with viruses. On the right, you can see a transmission electron microscopy picture of a virus only 100 nanometers in diameter. Which virus do you think it could be? The clue is the large knobby spikes that are sticking out of the virion's surface. If you thought coronavirus, you are correct. Viruses can also be identified by the effects they have on host cells. Pathogenic viruses can produce cytopathic effects, which you can see here in the pictures. The one on the left is a scanning electron microscopy image of healthy uninfected mammalian cells, which are nicely stretched out in monolayers, as epithelial cells tend to do when they're in culture medium. When I used to do tissue culture, we used to say that the cells must be happy since they're waving at us. On the right, you can see that they look very different. When a cytopathic effect is observed, cells curl up in round balls and bunch together. Usually, they'll die and lyse, so large holes can be seen in the cell monolayers when viruses are present. Plaque assays enable the amount of virus present to be quantified. Serial dilutions of the virus sample are added to cell lines in tissue culture wells. The areas of dead cells appear as clear areas called plaques. By counting these areas, the number of plaque-forming units can be determined, and the dilution factor used to calculate the original amount of virus, and this is called the titer. The diagram on the right shows a tissue culture plate with serial dilutions of SARS coronavirus 2, the virus that causes COVID-19. In this 6-well plaque assay plate, you can see that the negative control, which is the first well on the left in the top row, shows an intact monolayer stained purple. The next well on the top row has a 1 in 100 dilution of the virus sample added to it, and this is the highest concentration of virus. You can see that most of the cells have lysed. In the next well on the top row, the virus sample was diluted to 1 in 1,000, and there are more than 100 clear-colored plaques. On the bottom row, the number of plaques in the wells are small enough to be counted accurately. In the first well on the left, the dilution of the virus sample was 1 in 10,000, and there were 21 plaques observed. In the next well, in which 1 in 100,000 dilution of the virus was added, there were only 2 plaques. In the last well, the virus sample was diluted by 1 in a million, and no plaques were observed, indicating the absence of SARS coronavirus 2 plaque-forming units. The number of plaques in the well with the 1 in 10,000 dilution was used in the calculation of the virus titer, which was determined to be 2.1 times 10 to the 5, or 210,000 plaque-forming units per mil. The counts in the other wells were not used because they were too low and too high to be accurate. This method is very similar to counting bacterial colony-forming units on agar plates, which many of you perform in the etiology labs. The cell line used in this example are Vera cells, which are epithelial kidney cells that were obtained from an African green monkey more than 50 years ago. If you've never heard of these tests, I'll describe them now, as they're rather complex and confusing, so it may take some explaining. Normally, to look for the actual pathogen or the toxins it produces, we can do a cytotoxicity test by adding the pathogen or its culture supernatant to a cell line cultured in vitro. This is the first test above. If the cells are damaged, we can confirm that the toxin is present and the case is infected. However, in antibody neutralization tests, you really want to see the opposite because you're trying to detect the presence of antibodies that will neutralize the toxin or pathogen and prevent cytotoxicity from occurring. So if we look at the second test below, you can see that toxins are mixed with the antibodies, which will be the test serum from the probable case and the cell line. If there is no cell damage, it means that neutralizing antibodies are present, and the case is lab confirmed. If cell damage is observed, no neutralizing antibodies are present, and the case cannot be confirmed. Antibody neutralization tests are carried out because it is not always possible to isolate viruses directly from infected humans, or the infection has resolved and the virus is no longer present. To explain these tests in greater detail, let's look at real-life examples of a plaque reduction neutralization test. The tissue culture plate shows results from 2 different individuals whose serum samples have been pre-incubated with the Middle Eastern Respiratory Syndrome coronavirus at various dilutions. The row of 4 wells on the left is the test result from the serum specimen from the first individual, known as sample number 126, and wells on the right incubated with serum sample number 976 from the second individual. At the very top row are control wells with cells in them. You can see that no plaques are present in these wells because no viruses were added. In the next 2 rows below, 2 different dilutions of serum pre-mixed with virus were added to the wells. Almost complete neutralization was observed in the left well on the second row. The serum sample was diluted by 1 in 40, indicating that sample number 126 contained neutralizing antibodies to the MERS coronavirus. Sample number 976 also had antibodies, but the amount of titer was much lower because if you look at the 1 in 40 dilution, which is the third well on the right, 43 plaque-forming units were present as opposed to none for sample number 126. The wells on the very bottom row are also controls. These are actually negative controls, as virus samples which had the same concentration as the test wells were added to the wells without any serum. You can see many plaques present in both these negative control wells, and the numbers provide the baseline level to score the extent of antibody neutralization. If you look at the third well on the left, the 1 in 80 dilution of sample number 126, neutralizing antibodies were only present in very low or in insignificant amounts, with 32 plaque-forming units present, compared to 33 in the control. With sample number 976, more than 50% plaque reduction was observed, with the 1 in 20 serum dilution. This is the second well on the right, with only 22 plaque-forming units, compared to 56 plaque-forming units in the control well at the very bottom of the row. Once again, Vera cell lines were used in these tests. Aside from plaque assays and plaque reduction neutralization tests, Hemagglutination can also be used to measure either the amount of virus present or the antibody levels to them. Many human viruses have hemagglutinin, which are glycoproteins on their envelopes that enable them to aggregate red blood cells as well as bind to host cells. The influenza virus has many different hemagglutinin subtypes, which can change through a variety of mechanisms. Sometimes this can be a major mutation, especially if viruses from various animal hosts recombine. This may result in flu pandemics due to the sudden emergence of a completely new strain. Viral hemagglutinin sequences can also change gradually over time. That is why we need new seasonal flu vaccines every year. In the diagram, you can see how a virus that has hemagglutinin can bind red blood cells and aggregate them to form a visible precipitate. Hemagglutination can be a quantitative test to determine the amount of virus that is present. When red blood cells are agglutinated, they form a thin, dispersed layer at the bottom of the well. Serial dilutions of the virus can be prepared and added to the red blood cells in a series of wells. When no agglutination is obtained, the virus titer can be determined. In this example, the last dilution where agglutination can be seen is in the well with the 1 in 160 virus dilution. So this is the titer, as the well with the next doubling dilution shows no agglutination and the red blood cells form a tight pellet at the bottom of the well. With higher amounts of virus, more dilution will be required before agglutination is not observed. The same principle can be applied to hemagglutination tests and hemagglutination inhibition tests, which are used in the lab to diagnose viral infections. In the first test above, when a pathogenic virus is mixed with red blood cells in the absence of antibodies, hemagglutination occurs. However, in the second test below, when serum from a case infected with West Nile virus is added to the test, and if the case has neutralizing antibodies, as in the test below, no hemagglutination occurs as it is inhibited by the antibodies and the case is confirmed as positive. So just to go over the hemagglutination inhibition test again, this time using some test wells to make it more realistic. In the top row of the picture on the right, you can see several wells with test serum added to the red blood cells and West Nile virus hemagglutininin. The picture in the middle is the real-life picture of the fourth and fifth wells of the test strip in the top row. Agglutination is seen in the fourth well, indicating that the serum specimen from this case does not have any neutralizing antibodies to the viral hemagglutinin, and therefore the case does not have West Nile virus infection. This is a negative result. In the fifth well, we see the non-agglutinated red blood cells that have sunk to the bottom of the test well. Since there's no agglutination, the test result is positive, as neutralizing antibodies are present that are able to inhibit the agglutination reaction. This case is therefore confirmed by the lab test to be infected with West Nile virus. Let's move on to other tests that use the molecular properties of viruses. These use either viral proteins or nucleic acids to characterize or identify them. Even though viruses have small genomes, they have a number of proteins and genes which can be used either to detect them or differentiate them from other viruses. Many of these laboratory techniques involve the use of electrophoresis, which is a technique that separates biomolecules based on their charge. The diagram shows a vertical gel electrophoresis system, which has several wells at the top, in which samples can be added before running an electric current across the gel. This causes the samples to migrate down the gel and separate according to their charge and molecular weight. I'll go over this technique in more detail on the next few slides. One of the most common types of electrophoresis is called SDS-PAGE, which stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. In the picture, you can see a number of vertical gel electrophoresis systems. There is one closest to the left with a large tank, and in front of this tank, two gel casting apparatus. To the right is a smaller tank. The small systems are very useful for quick runs that provide initial data. On the shelf above is a power pack that provides electric current to run the electrophoresis system. I'm not sure what the laboratory worker is doing in this picture. Usually, the polyacrylamide gels are cast between glass plates, and because it is a polymerization reaction, the gels need a bit of time to set. It's a bit like making thin sheets of jello but in an upright position rather than flat. Then these are loaded into the buffer tanks before loading the specimens. Once this is done, the tanks are covered and the electrophoresis can proceed. Depending on the size of the tanks, this can range from minutes to hours. SDS page is used mainly for separating proteins. The principle behind the technique is the use of sodium dodecyl sulfate, which is a chemical that has a long carbon chain and negatively charged sulfate groups. The long carbon chains bind to the protein backbone and coats it with negative charges. When an electric current is applied across the polyacrylamide gel, the protein mixture is separated and travels from the negatively charged electrode at the top to the positively charged one at the bottom. The smallest molecule travels the fastest and ends up at the bottom of the gel. The opposite applies to the largest molecule. This travels the slowest and stays at the top of the gel. You can see the separation of two different mixtures of proteins in the picture. In lane number 1, a mixture of 6 proteins was separated. The smallest protein was 14.4 kilodaltons, and this is seen as the first band from the bottom of the gel. The other 5 bands are stacked on top of this smallest band, and you can see that the distance they traveled is dependent on their size. In lane number 2, a mixture of 3 high molecular weight proteins was separated. 2 of them are the same proteins also present in the first mixture, whereas the third protein is the largest one of all at 116.2 kilodaltons, and you can see the band at the very top of the separating gel. To analyze viral proteins, virus cultures are lysed and separated by SDS page. They can be visualized by staining the polyacrylamide gel using protein stains. However, because most viral lysates also contain mammalian cell proteins that are used in their culture, immunoblotting is usually performed after the initial electrophoresis to detect viral antigens using specific antibodies. Immunoblotting is also used for detecting antibodies to viral antigens. Before it can be carried out, the separated proteins need to be transferred to membranes which enable them to be probed more easily, as polyacrylamide gels, being gel-like in their consistency, can dry out with time and are quite difficult to store. The protein bands also start to diffuse once the current is switched off, so the gel needs to be fixed in alcohols, which can denature the proteins, causing antigenic properties to be lost. Since it's not possible to perform antigen-antibody reactions in polyacrylamide gels, proteins need to be transferred to membranes, which gives them better stability and allows them to be immobilized onto a thin sheet of membrane. This process of transferring proteins from gels to membranes is called Western blotting. This is a pictorial representation of a Western blotting apparatus. Once again, electrophoresis is used to push the protein bands in the gel to the membrane, which in this case is nitrocellulose. The assembly is made up of several sheets of sponges and filter paper with the gel and membrane sandwiched in the middle. It is important to orient the gel so that it is facing the negative electrode, otherwise the proteins will migrate out of the gel into the tank buffer, which means that they'll be lost forever. This picture shows a mixture of 9 proteins of varying molecular weights, which are used as molecular weight standards in Western blotting. Some of these molecular weight standards can be purchased in a pre-stained version, and some of them are stained in rainbow colors, so that you can see them separating right before your eyes on the gel, as well as monitor their transfer onto membranes. This is very convenient for timing coffee breaks, as it is important not to leave your SDS page gel running too long, since the lowest molecular weight proteins can drop off the bottom of the gel, and once again you lose them forever, which tends to be an unmitigated disaster, especially if you are particularly interested in the lower molecular weight proteins, and you spend a whole year or more growing and purifying viruses to get their proteins. Some of the markers also have special chemicals covalently bound to them so that they can be probed with the same reagents that are used in the immunoblotting procedure, such as biotin, which is used in the avidin-based enzyme detection reactions. I'll talk about that on the next slide. This is an immunoblotting tray which has many channels. It sits on top of a rocking apparatus. Once the western blot is complete, the membrane is cut vertically into small strips, about a centimeter wide. They're then placed in the channels, one strip per channel. They're then probed with antibodies using a series of steps that are very similar to those used in ELISAs or enzyme-linked immunosorbent assays. Just like the wells in ELISAs, once the proteins are transferred to the membranes, the first step is to block any free sites on the membrane to prevent nonspecific binding. The proteins are represented as yellow triangles. Usually, a protein solution such as skim milk or bovine serum albumin is placed in the channels and left overnight to bind to the remaining sites. These are represented as black discs. After the blocking reagent is removed, the membrane strips are ready to be used. The test serum, which is referred to as the first antibody, is added and incubated for an hour, after which the channels are washed to remove any unbound material, which may be non-relevant antibodies or proteins. The second antibody is then added. This is usually an anti-serum like anti-human IgG or IgM, and this second antibody is usually conjugated with a chemical such as biotin, which binds specifically to another chemical called streptavidin. Once again, the blot is washed thoroughly to remove any unbound second antibody. The next reagent that is added is the streptavidin conjugated enzyme, which will bind specifically to the biotin conjugated second antibody if it is present. In the final step, the enzyme substrate is added, and any proteins that are viral antigens will show up as dark purple bands. Let's look at a sample immunoblot. This one has 6 strips. The first 4 strips are test specimens using sera from probable HIV cases. The last 2 strips, that's lanes 5 and 6, are controls, with strip 5 being a positive control, probed with serum from a confirmed HIV case. You can see several bands on the positive control strip, which are diagnostic for the type of exposure and immune response of HIV cases. Strip 6 is the negative control, and it is blank except for a high molecular weight band at the top of the strip. For the first 4 test strips, you can see that the results are quite different. The first strip is negative, whereas the second strip is equivocal, meaning that it is difficult to tell, based on the results, whether the case has HIV. The third strip has a very strong band with the same size as one in the positive control, so the serum is likely from a case with HIV, but a second serum sample needs to be tested to confirm the result. Finally, the fourth strip is the most positive one of all. Even though it lacks some of the higher molecular weight bands, the serum sample is positive. Once large amounts of DNA are generated by PCR, genetic fingerprinting can be carried out to differentiate between different virus strains. This can be done using a technique called RFLP, or restriction fragment length polymorphism. Special enzymes called restriction enzymes cut DNA at sites that have specific DNA sequences. This means that for viruses with different DNA sequences, fragments with different lengths will be generated after treatment with restriction enzymes. These restriction fragments can be separated by gel electrophoresis, and the pattern visualized to see if it is identical or a variation. In the diagram, there are 2 different virus strains called A and B. They have slightly different sequences, and strain A has a minor sequence variation, which produces an additional third cutting site for the restriction enzyme. This restriction site is absent in strain B, as there is a DNA sequence variation in this portion of the gene. This means that strain B only has 2 restriction sites, and 3 fragments are produced, whereas 4 fragments are produced for strain A because it has 3 restriction sites. When these fragments are separated by gel electrophoresis, the RFLP patterns are different. 2 of the fragments from strain A are the same as those produced by strain B. These are the yellow and pink bands. However, strain A has 2 other fragments that are different, a smaller green band and a larger black band. The size of these 2 bands should add up to the size of the middle band in strain B. Gene sequencing or DNA sequencing of viral DNA is facilitated by the amplification of large amounts of DNA using PCR with specific primers that anneal to flanking ends on either side of the target sequence. Reverse transcription, or RT-PCR, is used for RNA viruses and PCR for DNA viruses. The PCR product is then sequenced using various techniques. The most common one is the Sanger or dye-terminated sequencing method. The Sanger or dye-terminated DNA sequencing method uses a primer that anneals to the target sequence. Reagents are added to the primer and template DNA that is being sequenced, including DNA polymerase, deoxynucleotide triphosphates, and a small amount of 4-dideoxynucleotide triphosphates, that's DDNTPs, that are labeled with fluorescent dyes in 4 different colors. The primer is elongated by the DNA polymerase. During primer elongation, the random insertion of a dideoxy NTP instead of a DNTP terminates synthesis of the chain at the 3' end because the DNA polymerase cannot add any more nucleotides due to the missing hydroxyl group on the dideoxy NTPs. This produces a series of DNA chains that vary in length by only one base. The DNA chains are separated by capillary gel electrophoresis, and the results are read by a computer imaging system. The sequencing results are usually visualized as a series of peaks in 4 different colors for the 4 different nucleotides A, C, T, and G. These show up as A in green, C in blue, T in red, and G in black, so the DNA sequence can be read easily. I've added a sample of the results that are generated. In this case, you're looking at part of the DNA sequence of the 18S ribosomal RNA gene of the protozoan parasite Cryptosporidium. You can see that it is quite an AT-rich sequence.